Today we'll take a look at a class of drugs that is not yet on the market outside of a clinical trial, the Bruton's tyrosine kinase inhibitors, Evobrutinib, Mesitnib, Tolebrutinib, Fenebrutinib. Why are people so excited about these drugs? We'll look at what is a tyrosine kinase anyway, and why do people think it has a special implication for certain people with multiple sclerosis? What are the potential side effects, and what are the results in clinical trials so far, and what clinical trials are ongoing? Remember what Charles Koch said, the only way to improve is to try new things. Now, we've certainly made a lot of progress with MS over the last several decades with disease-modifying therapies, and many people are doing well and having minimal side effects, but others have immunosuppression from highly effective disease-modifying therapies, and still others have progression despite the fact that the drugs are working in the sense they're not having relapses, not having new MRI lesions, they're having PIRA, progression independent of relapse activity. Why does this occur? Well, it could be due to neurodegeneration, but it could be due to smoldering inflammation. What you're looking at here is a stain from an autopsy study of microglia. These are cells in the central nervous system of the innate immune system, the immune system that you're born with, very different from the adaptive immune system, the B and T cells thought to be implicated in the initiation of inflammation in multiple sclerosis and the target of most disease-modifying therapies. But these have been seen in inactive progressive MS, so maybe progressive MS with inactive plaques isn't so inactive. It's just a slow, insidious, smoldering inflammation, and that may be why we often see worsening of existing deficits. And this has been associated with this slow progression, even with no relapses and no new MRI lesions. Can we see this in a living individual? Maybe the answer is yes. This is a 48-year-old woman with relapsing MS with an EDSS, Expanded Disability Status Scale of 4, moderate disability, still walking independently, had the disease for 12 years, and this is a specialized PET scan with a marker for microglia, and you can see there definitely is activity. So even in relapsing MS, she has this slow, smoldering inflammation that may be making her subtly worse and could drive a potential inevitable transition to secondary progressive MS. Can we see this on conventional MRI? Maybe yes in some people. This is a specialized scan looking at a paramagnetic rim lesion. You see this dark rim in a man in his late 50s with relapsing MS. These lesions have been associated with smoldering inflammation and progressive MS. And as you have already anticipated, yes, the Bruton's tyrosine kinase inhibitors do affect these cells of the innate immune system. But let's back up for a second. What exactly is tyrosine kinase anyway? So tyrosine is an amino acid residue of proteins. So it's a building block of proteins in the cell. Kinase is an enzyme that adds a phosphorus. So it's an enzyme that phosphorylates or adds a phosphorus to tyrosine residues of proteins. And as I'll show in a few slides, this initiates a cascade within the cell that changes the property of that cell. Which cells? Well, it's in mast cells and other cells of the immune system. Mast cells are involved in allergies. Importantly, microglia and macrophages, these are cells of the innate immune system implicated in smoldering inflammation, but also B lymphocytes, the target of various other disease-modifying therapies, Ocrevus, rituximab, Briumvi, Kesimpta, for example. And it influences their survival, their proliferation, and migration. So it doesn't kill these cells, it changes and regulates their activity. Now, importantly, this would be the first class of disease-modifying therapies for MS where it's an enzyme, and that could be very important because the individual drugs may make a difference because different drugs have different specificities for different tyrosine kinase enzymes, which may affect different cells differently. So we may see different efficacy and different side effects. Whereas with B cell depleters, if you kill the B cell, if you break it open and it's not there anymore, the individual drug may not matter as much. So that's one thing that I'm anticipating, a A side effect in an individual drug in this class may not be seen in other drugs within the same class, which is a little bit unique. Now, these tyrosine kinases are also in gastrointestinal cells, so gastrointestinal side effects are something we have to watch out for. And we have a lot of basic science studies on what these BTKIs do. They've been found to reduce memory B lymphocytes, which are pathogenic in multiple sclerosis, and they also modify the behavior of B cells. For example, they reduce certain receptors on the surface of B cells, such as cytokine and integrin receptors, such as VLA4, which is the target of Tysabri, and CXCR4. And BTKIs have already been used to treat other conditions. For instance, Mesitnib, which is being studied in MS, has already been used to treat certain types of tumors of mast cells in dogs. Trade name is Masivet. And Gleevec, imatinib, is perhaps one of the most famous drugs of all time, the first designer drug used to treat chronic myelogenous leukemia, and there are other BTKIs to use to treat various forms of hematologic cancer. Now, the most promising agents in multiple sclerosis, at least most likely to be approved relatively soon, are Tolebrutinib, Evobrutinib, and Fenebrutinib, and there are other agents that are a little early on in their development, such as Remibrutinib and Orelabrutinib. Now, BTKIs being used in MS, such as these three I've made red, are thought to be safer than other agents used for cancer because they're more specific and hopefully have actions in fewer cells more relevant to MS. For example, the drug Evobrutinib, used to treat certain immune cell cancers, also has effects on human epidermal growth factor receptor 2, HER2, which has been associated with cardiotoxicity or heart toxicity. Also, the effect on other protein kinases of non-specific binding can suppress T cells, and that could increase the risk of infections, whereas these have fewer effects, if any effects, on suppressing T cell function. And preliminarily, the risk of serious infections with BTKIs does seem to be lower than with, say, B-cell depleters like Ocrevus or Kesimpta, at least based on relatively short-term studies. Now, with longer-term studies, with continuous use over years, it could be a different story, but the advantage of these drugs is they, generally speaking, do not cause lymphopenia in most people or lowering of the white blood cells. I compiled some other side effects from various studies, including diarrhea, nausea, which makes sense because BTK is in the gastrointestinal tract, symptoms of inflammation or reactions such as swelling or rash, lethargy, sleepiness, anemia, sometimes elevation of the liver enzymes, generally not severe, and proteinuria or protein in the urine. And this picture just shows the tyrosine kinase pathway in general. And so you can see tyrosine kinase is a transmembrane enzyme. This is the cell membrane, and it phosphorylates these tyrosine residues, which in turn affects other proteins. And there's this very complicated cascade of events, but the end result is the change in the activity of the white blood cells. It doesn't kill them, it affects their proliferation, their differentiation, their survival, their migration to different tissues. And you have to understand there are different tyrosine kinases in different cells, so the specificity of the specific drug and its differential action in different cells could make a significant difference and again affect the safety and efficacy of the drug. This picture just shows the effect of BTK in multiple sclerosis theoretically. So for instance, BTK affects the expression on B lymphocytes of integrins. So if you block that, you may block migration of B cells into the central nervous system, similar to the action of Tysabri. We spoke about the microglia causing this low-level inflammation within the central nervous system, which does have BTK activated normally. Also, some people with progressive MS have follicles within the meninges, the coverings of the brain that contain these B cells. It would be interesting to see if these BTKIs affect these follicles. There's a prior study with intrathecal rituximab, giving the drug rituximab directly into the cerebrospinal fluid, which was not effective, and this was thought to be a marker of individuals who might benefit from that therapy, but hey, BTKIs are a different drug. So before we move to individual agents, I want to summarize the potential advantages BTKIs have over B-cell depleting drugs such as rituximab, Ocrevus, Kesimpta, and Briumvi. One is I think these drugs are safer. I think they likely have a lower risk of weakening the immune system and causing serious infections, and some people just aren't great candidates for immunosuppressants. Maybe they're older, sicker, just too prone to infection, or they've had serious infections. Related to that, these drugs likely have a faster offset, meaning you stop the drug, the effect goes away. So if you run into problems like an infection, you can just turn it off. Whereas if you take rituximab or Ocrevus for a long period and you have zero B cells and very low immunoglobulins, you can't just turn that off and make it go away. And sometimes it can take years for the immune system to return to normal. Now, the last thing is, even though I think BTKIs are less strong overall in terms of stopping relapses and new MRI lesions, they actually could be more effective in people with smoldering multiple sclerosis. Because as we've seen in various studies, the effect of stopping new lesions isn't all that highly correlated with stopping disability progression in the long run. So maybe these drugs could even be better and safer, although we would only know that for sure with a head-to-head trial. Next, we'll move to the individual agents, and I'll show you just a little bit of data for each drug. So we'll start with Mesitnib, and this was the first published trial in progressive MS with a BTKI. And this is a phase 3 trial, a randomized double-blind controlled trial, and they looked at primary progressive MS or people with secondary progressive MS who were relapse-free, and they used 2 different doses of Mesitnib, 4.5 milligrams per kilogram per day or 6 milligrams per kilogram per day, given twice daily, divided twice daily. This is an oral treatment, a pill. And there were 656 participants, and they took people up to age 75, which is really good. I really want to see how this drug works in older people with progressive MS, especially people with more disability. The median age was 50. The inclusion criteria required that you be able to walk with a cane or better. In other words, if you needed a walker to walk, you could not get into the trial, which is very unfortunate in my opinion. And the primary outcome was changes in the EDSS, a measure of disability in MS. Now we'll look at the data for the 4.5 milligram per kilogram per day group, and they used the least squared mean change of the EDSS of each data point. In other words, the 12-week data point, the 24-week data point, this 36-week data point, they took them all, which is a little bit strange. You would think they would just look at the very end of the study, but they specified this in advance. So you can see that the placebo group in green gets worse, and the Mesitnib group got a little bit worse, but not as much worse. And if you took the average of all of these data points, people getting Mesitnib only worsened by 0.001. Now you can see they were worse at the end, but they were a little bit better at the beginning. And so essentially, on average, there was no change. Whereas with the placebo, the average change was 0.098, or approximately 0.1. And so there was a difference. It was statistically significant with a p-value of 0.0256, but the absolute difference was very small. A 0.1 difference on the EDSS scale is not a very large difference. I'm not sure that's clinically significant if this drug causes significant side effects. Another thing is that the 6 milligram dose was completely ineffective. You can see the placebo group actually got better at the beginning and then maybe a little bit worse, but no statistically significant difference. And I presume this is the reason we're not seeing further trials with Mesitnib, because the data just weren't that impressive. But what about Evobrutinib? This is another oral BTKI. It's irreversible, meaning when it touches the enzyme's dead, and even after the drug goes away, it can't be reactivated. It can only be replaced by the cell. The half-life of the drug itself is very short, only 2 hours. How long is the biological half-life? I honestly have no idea. And what you're looking at is a phase phase 2 trial in relapsing remitting MS, not progressive MS, and they compared placebo to different doses of Evobrutinib and against Tecfidera or dimethyl fumarate. So they looked at 25 milligrams once a day, 75 milligrams once a day, and 75 milligrams twice a day. And you can see there's this dose-dependent response. The primary outcome was the total number of enhancing or gadolinium active lesions over a 24-week study. And you can see that the Evobrutinib 75 milligram twice a day dose did the best. A couple things to note here. One is that Tecfidera somehow did worse than the placebo group, even though it has previously beaten placebo in randomized controlled trials. Why is that? I don't really know. This was an open label arm, and obviously people entered the study hoping to get Evobrutinib, so maybe they were so disappointed and stressed out when they got randomized to the Tecfidera arm that they didn't even take the drug. I have no idea. And you can see the average number of enhancing lesions with placebo was around 3.85. So low dose Evobrutinib did nothing, no change. 75 milligrams once a day reduced lesions by around half, and Evobrutinib 75 milligrams twice a day reduced lesions, enhancing lesions to an average of 1.15, which is an improvement, but honestly not that great. This is a less than 75% reduction in gadolinium enhancing lesions, and this would correlate more with a low to moderate efficacy drug, not a high efficacy drug, which may reduce gadolinium enhancing lesions by over 95% easily. So it doesn't look that good at suppressing active lesions. They did look at some markers of smoldering inflammation, looking at slowly expanding lesions, and they found that there was an effect with Evobrutinib. So maybe lesions aren't everything. They also looked at relapses, but this was a very small study, so it's really hard to draw conclusions. If you look at the 75 milligram twice a day dose, which is the dose that will now go on to phase 3 trials, the reduction in relapses, the relative risk was 0.23. In other words, a 77% reduction in relapses, but it actually was not statistically significant. You can see the confidence interval crosses 1, but this effect size would be very clinically significant, and you can see 0.08 annualized relapse rate, relapses per person per year, or around 1 relapses per every 12 years per person, is very good compared to the placebo 0.37, around 1 relapse every 3 years, so it probably suppresses relapses to a decent extent. But Tolebrutinib may be more attractive. It's a once-a-day pill, also an irreversible BTKI, and it's reported to have good penetration into the central nervous system in humans within 2 hours of one dose. I'll show you that data in a moment. The half-life of the drug is very short, again, only 2 years. How long is the biological activity of the drug? I have no idea. What you're looking at is data from a 16-week phase 1b double-blind randomized trial, age 18 to 55, relapsing remitting MS and active secondary progressive MS, and it looked to be a little bit better than Evobrutinib. This is data on new T2 lesions. They looked at placebo versus different doses of Tolebrutinib, most of which were ineffective, but the 60 milligram dose looks good. You can see 0.23 new T2 lesions versus 2.12. That's an 89% reduction, pretty good. If you look at enhancing lesions, there were 93% fewer active lesions with the 60 milligram dose compared to placebo. Again, the lower doses was not effective, so you really need that higher dose, and this is the dose that's being studied in phase 3 trials. Now, they actually did a study to look at CNS penetration, and they gave people a single 120 milligram Tolebrutinib dose and did a spinal tap, and they were able to detect a decent amount within the cerebrospinal fluid just 2 hours later. Bless the souls of the people who subjected themselves to an unnecessary spinal tap just for the sake of science. The side effect profile preliminarily looks good. These are side effects with different doses of Tolebrutinib. Focus on the 60 milligram column here. You can see there was one serious adverse event. This was actually a multiple sclerosis relapse requiring hospitalization, not necessarily necessarily a side effect of the drug. Upper respiratory tract infection, nasopharyngitis, same thing, no diarrhea in the higher dose. There was one person who had elevated liver function tests, and this has also been reported in the ongoing phase 3 trial, so that's something to look out for with Tolebrutinib. For Fenebrutinib, we don't have any published data, but this is a press release from Genentech, the developer of this drug. Fenebrutinib is an oral reversible BTKI, not irreversible. Does that matter? I'm not sure, but I'm mentioning it just in case. It's also been studied in other autoimmune diseases such as rheumatoid arthritis and lupus, and it has a slightly longer half-life of 4 to 10 hours as opposed to 2 hours for Evobrutinib and Tolebrutinib. So in their press release, they say that this drug in a relapsing MS trial reduced active MRI lesions, a phase 2 trial, and the p-value was 0.0022. Is that p-value so good due to a large effect size or a huge sample size? I'm not sure, but more details to follow very shortly. And there are tons of ongoing trials as well. These are some of them. For Evobrutinib, there are 2 phase 3 trials for relapsing MS. Both of them are head-to-head trials against Aubagio, a low-efficacy pill for multiple sclerosis. You may be quick to criticize the drug companies saying they're selling out. We already have good drugs for relapsing MS. But most of the progression of disability, even in relapsing MS, is PIRA, progression independent of relapse activity. So it is possible these drugs are better than existing drugs. Now, am I going to prescribe them to a young person with aggressive relapsing MS with no contraindication to taking immunosuppressants? Absolutely no chance unless they actually did a head-to-head trial against B-cell depleters. But hey, in the distant future, it could be possible. And again, some people just aren't good candidates for immunosuppressants. For Tolebrutinib, in relapsing remitting MS, there's a head-to-head trial against Aubagio, 2 such trials, Gemini 1 and Gemini 2. Also, a study in non-active secondary progressive MS against placebo, Hercules, and in primary progressive MS versus placebo, Perseus, which gets a ton of hype, and I have 2 patients who entered this study, though they do have the problem with the elevated liver function test, so we'll have to keep an eye on that. But that is the one trial I'm really keeping a close eye on. Fenebrutinib, 2 trials in relapsing MS against Aubagio, and 1 trial in primary progressive MS, Fentrepid. And the other 2 drugs are maybe a little earlier on. Remibrutinib, 2 relapsing MS trials against Aubagio, and Orelabrutinib, a phase 2 trial against placebo. So I'd be interested to know, what do you think about these BTKIs? Would you change to this drug? Have you entered these clinical trials? And do you have suggestions for future videos?